Unit is about cell division. Cell division is incredibly important. So why do cells divide? They divide for repair, growth, or development. If you break a bone, you need more cells to fuse and heal that bone. If you are a growing teenager, you need more cells to continue to grow. And if you are developing in any way, you need more complex cells that are going to need to continue to make more cells. In order for cells to divide, they need to go through something called the cell cycle. There are 4 phases of the cell cycle. It will go G1, S, G2, and M. Interphase consists of those first 3 phases, in which case G1 you'll experience growth amongst the cell, S you will have the DNA copy itself or DNA synthesis, and then G2 you'll have more growth and you'll prepare for mitosis. That 4th and final phase, the M phase, is mitosis or meiosis, but is about the division of the nucleus. In normal somatic cells or body cells, skin cells, heart cells, liver cells, you're going to experience mitosis, in which case prophase is going to dissolve the nucleus. Metaphase, you're going to line up these structures called chromosomes along the equator or center of the cell. In anaphase, you'll pull them apart, and in telophase, you'll reform a nucleus around either side of those. Then you will go through cytokinesis, in which case the cell will split into 2 cells. In plant cells, we will need a cell plate or these large boxy looking structures in order to create a cell wall. No matter what, whether you go through cytokinesis in animals, cytokinesis in plants, the result is 2 identical daughter cells through mitosis. When you go through meiosis, the process is a little bit different. The reason that the process is different is because in meiosis, you're producing gametes. Gametes are sex cells, either sperm or egg cells, and they're the end result of meiosis. They're not an ongoing process. Haploid refers to the number of chromosomes within those sperm and egg gametes. The reason they have half the genetic information is because eventually they'll go through a process of fertilization or the unification of gametes. So you take sperm, you take egg, both with half the genetic information. They combine, now they have the full amount of genetic information. Diploid cells refer to mitosis cells. Every other cell is diploid, has 46 chromosomes, or twice the number of chromosomes. It's going to be what's in body cells. So meiosis is incredibly similar to this process with a few key differences. We will go through meiosis twice. That's why we have meiosis 1 and meiosis 2. The reason that we do this is to get the end result of 4 unique haploid cells. So in prophase 1, yes, the nucleus will dissolve, but another event called crossing over will occur, where homologous chromosomes or similar chromosomes next to each other will trade pieces of their DNA at the very end. In metaphase 1, we'll line these chromosomes up in pairs. In anaphase 1, we'll separate them by entire chromosomes. Instead of chromatids, the entire chromosome is separating. Telophase 1 will reform nucleus on either side, split. Then in prophase 2, the reason there are 2 cells is at every step of meiosis 2, you are having 2 cells go through this process. In prophase 2, it's identical to prophase, it's just happening in 2 cells. In metaphase 2, it's identical to metaphase, it's just happening in 2 cells. Anaphase 2, separate the chromatids, and then in telophase 2, you're reforming the nucleus around 4 unique haploid cells. That is meiosis, that is mitosis. For both of them, though, we need to make sure that we are monitoring throughout. What happens is we have checkpoints at G1, G2, and metaphase that are enacted by cyclins and cyclin-dependent kinases. These are what regulate the cell cycle. A popular one of these is P53, which is a tumor-suppressing gene which, when mutated, causes cancer. The reason that these checkpoints are so important is we can tell a cell to go to cell arrest or stop dividing. We can tell a cell to go through apoptosis or programmed cell death, preventing it from continuing to divide. If we fail to do that, that is when cancer happens, which is unregulated cell growth of cells. If you have a bunch of healthy cells inside your arm, and then a few of those cells go bad, they will continue to take over and continue to grow until they're clouding the nearby area. And in fact, sometimes they can grow outside of the skin or outside of your normal area, forming a tumor or a mass of cells. Once cancer has occurred, there's no treatment to it, but there are certain things that we can do. Surgery is the removal of a tumor, chemotherapy is the flushing of chemicals to kill multiple cells, and radiation is using UV rays to try and kill more of these cells. That is cell division in a nutshell, and that should be it. You guys should be good for the test. Cool.